So let's now talk about the regulation of glycolysis. And before we start into these details, let's just revise the reactions of the pathway quickly. So we start with the glucose molecule, and then we phosphorylate it to glucose 6-phosphate, okay? And then we isomerize it to fructose 6-phosphate. And then we add another phosphate to become fructose 1,6-bisphosphate. After that, we cleave this molecule into two molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. In the next step, several changes happen until we reach the formation of phosphoenol pyruvate. And then phosphoenol pyruvate is converted to the last product in this pathway, allyl pyruvate. An important thing that we should keep in our mind that most of the reactions of glycolysis are reversible. Yani, if you look at the reactions that we discussed last time, you'll see the arrows of the reactions going both ways, forward and reverse. Okay, and the enzyme can catalyze the reaction in the forward direction and reverse direction except for 3 steps. شو هم هدول الsteps? The first one is the phosphorylation of glucose by glucokinase or hexokinase. Okay, the second one, the phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate by phosphofructokinase. The last one is the formation of pyruvate from phosphoenol pyruvate. تمام؟ فهدول الثلاث خطوات they are irreversible. They are catalyzed by enzymes that only catalyze or that are only able to catalyze the forward reaction. The reverse reaction can be, or these reactions can be reversed by other enzymes. Okay, since they are irreversible steps, so they are the steps at which regulation happen. Okay, يعني أخذتوا قبل هيك خصائص الخطوات اللي بتكون irreversible منها committed steps أو rate limiting steps إلى آخره. فهدول الخطوات الثلاثة هم اللي فعليا بصير عليهم عملية الريجوليشن، فرح نحكي regulation for each of these 3 reactions. تمام؟ طيب in this figure we're showing the regulators, whether they are positive يعني activators or negative inhibitors, negative regulators or inhibitors for glycolysis and for gluconeogenesis. We're not going to discuss into details the regulators of gluconeogenesis now, but just keep in your mind that gluconeogenesis is the pathway or the opposite pathway of glycolysis. يعني glycolysis, we break down glucose to pyruvate. In gluconeogenesis, we use pyruvate and other molecules to synthesize glucose. Okay, so we expect that if this is an activator of glycolysis, it would be an inhibitor of gluconeogenesis and vice versa. يعني هيك general, this is the general scheme, but there are some other details that we'll discuss later for gluconeogenesis, but now we're just going to talk about the glycolytic regulators. تمام؟ طيب هلا for fructose 6-phosphate conversion to fructose 1,6-bisphosphate by phosphofructokinase, there is the fructose 2,6-bisphosphate مش 1,6 اللي شفنا له البرودكت. 2,6 is an activator. AMP is an activator as well. Okay, ATP is an inhibitor. Citrate is an inhibitor. Proton is an inhibitor. تمام؟ هلا بنحكيهم احنا هيك ذكر بس كمان شوي بنشرح شوية تفاصيل. تمام؟ generally يعني أو من اللي حكيته هلا you can يعني quickly understand why AMP is an activator. AMP reflects a low energy state in the cell. Okay, that's why I would expect if there is a high concentration of AMP, there would be activation of the pathways that produce energy of these glycolysis. Okay, ATP on the other hand is a marker of high energy state. So if I have ATP already, why do I break down glucose to get energy? Okay, so this is just very quick and we'll go into the details of others later, inshallah. For the third reaction اللي هو الphosphoenol pyruvate to pyruvate reaction catalyzed by pyruvate kinase. This enzyme is regulated by the activator fructose 1,6-bisphosphate. This one is going to act as if there is a high amount of it, is going to activate the next steps to specifically this one, this regulated step. ATP again is an inhibitor, and this makes sense for you because it's a reflector of or marker of high energy state in the cell. And alanine, and we'll see later on inshallah why alanine is an inhibitor of this pathway or of glycolysis, specifically for pyruvate kinase. Let's start first with the first enzyme, the glucokinase or hexokinase. And remember, we mentioned that this first step can be catalyzed by the two enzymes, glucokinase and hexokinase. Glucokinase is specific for glucose and is an induced enzyme. So there has to be a high concentration of glucose for it to get activated. Okay, and this high concentration of glucose is going to be uptaken by or induced. Their uptake is going to get induced by the secretion of insulin. Okay, so let's look at this curve that takes the or plots the glucose concentration with the enzymatic activity for both glucokinase and hexokinase. Notice that hexokinase is, we said that it's active all the time. We don't have to have a very high concentration of glucose to get it activated. So very small amount of glucose is going to activate it more than the glucokinase, as you can see in here. Then it's going to reach its Vmax. And Vmax of this hexokinase is much lower than that for glucokinase. And that makes sense because glucokinase is activated when in a in a well-fed state, where you took a high carbohydrate diet or a carbohydrate-rich diet. So we need to get this enzyme activated so we don't have the glucose accumulating and increasing in the cells of the person or of your cells after the consumption of this high or glucose-rich meal. Okay. Notice also that from the curve you can tell the Km value. So Km value, remember from last time we revised it. It's the concentration we need to saturate half the amount of the enzyme, so, or to reach the half the Vmax of the enzyme. Okay, so this is the Vmax. This is half of it. Okay, almost half of it, and this is the Km value. Sorry, this is the Km value here. Whereas for hexokinase, this is the Vmax here. So it's something like here. This is the Km. Notice that the Km values are very different, and we commented on that last time. So this is the Km for hexokinase. It's much lower, reflecting the higher affinity for glucose and detection and binding of glucose at very low concentrations. Whereas for glucokinase, as you can see in here, yeah, we need a higher concentration for to reach Vmax over 2. Okay, so this is just to show you what happens in normal situation. There's no regulation so far, but the and this is and this orange region right here. This is the concentration of fasting blood sugar. So you can see that at fasting or after 2 hours after a meal, we will reach this, or we will have more activation of the hexokinase, and it reached its maximal velocity or the Vmax, whereas it's still much smaller, much lower for glucokinase, because at fasting blood glucose levels we wouldn't have a high concentration of glucose like after a meal. Okay, فإذا بيختلفوا عن بعض. في خلال بعد الوجبة مباشرة ما راح يكون هيك المستوى، راح يكون أكثر الشجر، فبالتالي there would be activation of the glucokinase. لو ما كان في meal، يعني الفاستنج blood sugar at least 2 hours after a meal، راح يكون عندي زي ما انتم شايفين الهكسوكاينيز reaching the maximal velocity وشغال وactive، في شوية activity من glucokinase but it's much lower than that for hexokinase. طيب خلينا نشوف glucokinase regulation as a specific enzyme for glucose. So whenever a person, for example, took a high carb or a carbohydrate-rich meal, تمام, there would be a secretion of insulin. Insulin is going to increase the amount of glucose transporters on the cell membrane, so the cells can uptake more glucose into the cell. More glucose in the cytosol would activate glycolysis, okay, to glucose 6-phosphate, to fructose 6-phosphate, etc., until we reach pyruvate. The presence of high concentration of fructose 6-phosphate that would end up in the production of pyruvate, and then pyruvate to acetyl-CoA and acetyl-CoA to Krebs cycle and production of high amount of energy. At some point, if there is a very high amount of this fructose 6-phosphate, it would act on glucokinase enzyme by sequestering it, يعني sequestering يبعده, and taking this enzyme to the nucleus. In the nucleus, there is a glucokinase regulatory protein, GKRP, to which this glucokinase is going to bind. Once it's bound to this regulatory protein, it becomes inactive. يعني صار عندي كتير من الفركتوز 6-phosphate راح يعطيني كمية كبيرة من البيروفيت، فبالتالي no need to continue these reactions. فlet's inhibit glucokinase. On the other hand, whenever we have اللي هم الفيرست steps, whenever we have high concentration of glucose, this high concentration of glucose is going to activate the release of glucokinase from nucleus to the cytosol, so it can activate a series of reactions. يعني أول ما يدخل الجلوكوز concentration عالي منه راح يرتبط في الجلوكوكاينيز، راح ينشط الجلوكوكاينيز by removing or separation from the regulatory protein in the nucleus and release into the cytosol to activate the reactions. Whenever we have, or once we reach to the point where we have high concentration of fructose 6-phosphate, this regulatory molecule, fructose 6-phosphate, is going to bind or is going to sequester the glucokinase to the nucleus to bind the inhibitory regulatory protein. Let's talk about the regulation of other enzymes like phosphofructokinase, for example, and the ATP molecules and AMP as well. So let's compare these different states. We have the exercise state, whenever you are exercising or you are resting. Same thing for the three molecules ATP, ADP, and AMP. Whenever we have a high concentration of the ATP, okay, so this means that we are in a high energy state. Whenever we have a high concentration of AMP, this means we are in a low energy state. Okay, notice that in exercise, when you are exercising, notice that the concentration is going to get low. بنزل concentration when you are exercising. For ATP, concentration of ATP is going to get lower. إشي طبيعي لأني أنا عم بستهلك منه بحتاجه للهذا، مقارنة طبعا مع الريست. For other molecules, notice that their levels generally are lower than that of ATP. تمام، هاي طبعا احنا بنعلق في هذا، او خليني ارجع شويه لورا، بنعلق او بنربط مع الفيجر هون اللي بيحكي لي عن الريجوليترز اللي هم AMP and ATP right here, but specifically أكثر للphosphofructokinase since it responds to both AMP and ATP. Okay. So whenever we have high concentration of ATP, so it reflects in the high energy state, this would inactivate the enzyme. Whenever we have a higher concentration of AMP, there would be actually activation of the enzyme because it reflects a low energy state. خلينا نيجي لشوية هيك على الكيرفز بشكل أفضل وspecifically الphosphofructokinase that is regulated by other molecules. And of the important regulators, positive regulators, اللي هو الفركتوز 2,6 bis phosphate. تمام؟ هلا مش 1,6 يعني مش the product of the reaction, it's another molecule اللي هو الفركتوز 2,6 bis phosphate. لاحظوا كيف الكيرف this is the fructose 6 phosphate اللي هي عبارة عن reactant of this reaction and this is the V over V max. طيب خلينا نشوف لما this is the normal curve يعني بدون إضافة أي شيء. Whenever we add fructose 2,6 bis phosphate or AMP اللي هم positive regulators, notice that we reach the Vmax, okay, at a lower concentration of fructose 6 phosphate. يعني they're going to act as positive regulators. طيب. This fructose 2,6 bis phosphate is produced by another phosphofructokinase. يعني هلا الphosphofructokinase if you remember converts or phosphorylates fructose 6 phosphate to fructose 1,6 bis phosphate. This enzyme is called phosphofructokinase number 1. There is another enzyme اسمه phosphofructokinase number 2 that produces fructose 2,6 bis phosphate. The importance of this molecule, fructose 2,6 bis phosphate, that it acts as an activator for glycolysis, as we see here, and it also acts as an inhibitor for gluconeogenesis. تمام؟ يعني لما يكون في عندي activation of glycolysis, I need to turn gluconeogenesis off. يعني إذا أنا عم بكسر الجلوكوز ليش أصنعه؟ صح؟ يعني هيك بصير عمليات متضادة وفيها هدر لطاقة الخلية. فإذا أنا بحتاج أصنع الجلوكوز بصنع الجلوكوز وما بكسره، يعني بعمل inhibition للجلايكوليسيس and activation of gluconeogenesis. إذا أنا عندي جلوكوز كتير بكسر الجلوكوز وما بصنعه بالجلوكونيوجينيسيس. تمام؟ فعشان تخلوها واضحة. فعشان ما أدخل بهاي هيك الحلقة المفرغة اللي ما منها أي فائدة, this one is going to act as a regulator for both glycolysis and gluconeogenesis. بنخليهم بالاتجاهات المتعاكسة. يعني activation of glycolysis and inhibition of gluconeogenesis. For this reaction اللي هو activated by phosphofructokinase, in which fructose 6 phosphate is converted to fructose 1,6 bis phosphate, there is another substrate. يعني we need the ATP as a source of the phosphate group. Notice that again same thing. This is the other substrate. يعني غيرت بدل من fructose 6 phosphate حطيت ال ATP and again V over V max عم برسمها. لاحظوا whenever I have just يعني normal reaction, the velocity V over V max increases until it reaches the maximum, then it goes down حتى لو أنا بزيد كمية ال ATP. تمام؟ Because يعني this is also dependent on the availability of fructose 6 phosphate اللي هي معتمدة على availability of glucose. Okay? طيب whenever I add fructose 2,6 bis phosphate اللي هو ال positive regulator or AMP again is a positive regulator, notice that at higher concentrations of ATP still we have higher activity of the enzyme. تمام؟ فهذا يعني بنرسم الكيرف نفسه اللي شفناه بالسلايد اللي قبلها بحالة إنه أنا يعني مثلتها نسبة إلى ال other substrate اللي هي ال ATP that we need or that we use in this reaction. طيب نيجي للإنزيم الثالث اللي بصير عليه regulation اللي هو ال pyruvate kinase which converts phosphoenol pyruvate to pyruvate. ال positive regulator اللي هو الفركتوز 1,6 اللي هو العادي اللي ضمن الباثواي 1,6 bisphosphate. النيجاتيف اللي هو ال ATP. Again that makes sense if I have ATP why I would break down glucose to get ATP. فبالتالي the whole pathway is inactivated. والثird one or the second negative regulator اللي هو الألانين. هلا الألانين ده just briefly and you'll know this more إن شاء الله later on, but just briefly الألانين amino acid once metabolized it gets converted to pyruvate. يعني if I just take the amino group of alanine out and I remove it, the rest of the molecule is actually pyruvate. The alanine is a source of pyruvate. So if I have plenty of alanine, I can get pyruvate. So no need to synthesize it via glycolysis. That's why it acts as an inhibitor for this specific step that produces pyruvate. Let's now talk about the hormonal regulation. احنا حكينا عن المولكيولر أو molecules that act as activators or inhibitors by يعني acting specifically on the irreversible steps or the enzymes of the irreversible steps. So how are these regulated or glycolysis is regulated in terms of hormones? يعني كيف بيأثر الهرمونات على الpathway and where do they act? Again, the most important hormones related to this pathway هم insulin and glucagon. And you all know that they act in opposite ways. So if there is high amount of insulin, there would be low amount of glucagon. And you all know that insulin is secreted after a carbohydrate-rich diet. تمام؟ والglucagon طبعا بيكون قليل. So glucagon can be secreted, for example, at fasting conditions. بالعكس لما ما في عندنا سكر كتير في الدم. Again, these hormones, two hormones are going to act on the irreversible steps اللي هم تبعت الجلوكوكاينيز الفوسفوريليشن. Again, phosphofructokinase, phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate, and the phosphoenol pyruvate conversion to pyruvate by pyruvate kinase. هلا الانسولين we would expect it to be ايش؟ a positive regulator. ليش؟ لأنه في عندي جلوكوز كتير when we take a carbohydrate-rich diet, there would be absorption of this glucose, high glucose level in the blood, more secretion of insulin, more expression and higher concentration of glucose transporters on cell surfaces or on cell membrane, more uptake of this glucose into the cells. So this would, more uptake of this glucose, معناته the concentration of glucose in the cells is high, so this would activate glycolysis. Okay, specifically at these steps. But glucagon is going to act the opposite way. It's going to be an inhibitor for these steps. نحكي هيك شوية تفاصيل وكيف بيدخلوا الهرمونز في عملية الregulation, and we'll take phosphofructokinase as an example. So, and we're talking about a well-fed state or after a carbohydrate-rich diet. There would be high concentration of insulin, and remember that insulin binds to receptor tyrosine kinases. These are the insulin receptors. Whereas glucagon binds to glucagon receptors, which are G protein coupled receptors. At this condition, well-fed state, there would be high concentration of insulin that binds to its receptor, and there would be low or there would be no glucagon that can bind to the G protein coupled receptor. طيب هلا نتيجة لهالكلام there would be activation of this pathway underneath receptor tyrosine kinases and activation of so many enzymes that would activate glycolysis and other pathways as well. Whereas underneath the glucagon, because there's no activation, there would be no formation of cyclic AMP by adenylyl cyclase. Okay, so there would be a lower activity of protein kinase A, because if there is cyclic AMP, there would be activation of protein kinase A. There is no cyclic AMP, there would be no activation of protein kinase A. Okay, طيب there's no activation of protein kinase A. What would happen? معناته هذا الكاينيز is not going to phosphorylate its targets. Of these targets اللي هو phosphofructokinase number 2. حكينا لكم إنه في عنا phosphofructokinase number 1 which acts in glycolysis, and there is another one phosphofructokinase number 2. This enzyme, or phosphofructokinase number 2, has two activities. It has a kinase activity on one side and it has a phosphatase activity on the other side. Okay. Once there is no phosphorylation, okay, إذا ما في phosphorylation, so the enzyme is dephosphorylated. So what would happen in this case? If the enzyme is dephosphorylated, the phosphofructokinase is active and the phosphatase is inactive. So they have to be in opposite يعني activity states. ما بصير يكون kinase وفوسفاتيز بنفس الوقت شغالين. ففي حالة إنه dephosphorylated, the kinase is active and the phosphatase is inactive. لو كان في glucagon يعني in low energy states, in low sugar states, there would be اللي هو activation of formation of cyclic AMP, activation of protein kinase A, بعدين phosphorylation of this. Phosphorylation would result in the inhibition of or inactivation of the kinase and activation of the phosphatase. اوكي هلا نحكي عن الوضع اللي احنا فيه اللي هو الهاي كربوهيدرات دايت او كربوهيدرات ريتش دايت. So the kinase is active. اسمه phosphofructokinase number 2. It acts on fructose 6-phosphate to become fructose 2,6-bisphosphate مش 1,6 2,6-bisphosphate. وحكينا انه الفركتوز 2,6-bisphosphate is a positive regulator of phosphofructokinase number 1 that converts fructose 6-phosphate to fructose 1,6-bisphosphate and activate طبعا reactions of glycolysis. Okay. طيب لو كانت الحالة بالعكس, glucagon is high, it's going to bind when there would be no binding, activation of adenylyl cyclase, cyclic AMP formation, activation of protein kinase A. Protein kinase A is going to phosphorylate زي ما حكينا. Phosphofructokinase number 2 is inactive, so there would be no formation of fructose 2,6-bisphosphate, whereas the phosphatase is active. بالعكس الفوسفاتيز يعني بيشيل الفوسفات من الفركتوز 2,6-bisphosphate and converts it back to fructose 6-phosphate. Okay. Let's talk about the hormonal regulation of pyruvate kinase اللي هو third enzyme that is regulated in this pathway. So this is the glucagon, as you know. This is secreted under low sugar states at fasting conditions, let's say. Glucagon is going to bind the G protein coupled receptor, activates adenylyl cyclase, so the ATP is converted to cyclic AMP. Cyclic AMP activates the protein kinase A, and again, as you remember, protein kinase A has so many targets. حكينا واحد منهم phosphofructokinase number 2. There's another target for it اللي هو pyruvate kinase. Once this pyruvate kinase is phosphorylated, it becomes inactive. So this would inactivate or inhibit the conversion of phosphoenolpyruvate to pyruvate, يعني بيعمل inhibition of glycolysis. يعني هذا الانزيم لما بيكون phosphorylated it's inactive. Once it's dephosphorylated it becomes active. وهي الحالة اللي بتكون في حالة الانسولين. لما يكون في insulin high concentration there would be no binding, فكل هذا الباثوي the whole pathway is inhibited. There would be no phosphorylation of pyruvate kinase, so it becomes active and continues the last step of glycolysis. طيب خلينا ناخذ a clinical hint or a clinical application on this, specifically on pyruvate kinase اللي هو the last enzyme in glycolytic pathway, and actually the most commonly affected enzyme by deficiencies in glycolysis. وهو أكثر enzyme بيصير فيه deficiencies بهذه pathways. An important thing that RBCs specifically cells, they're mostly affected by this deficiency. ليش؟ خلينا نشوف شو بيصير في عند هدول patients. هلا هاي deficiency it may for example result from reduced enzyme activity or reduced amount of the enzyme or changes in the characteristics اللي هي الانزيمولوجي أو الانزيماتيc characteristic like the Km, the Vmax, etc. due to abnormalities or due to for example genetic mutations that result in structural changes in this enzyme as a protein. تمام؟ فصار في تغيير. المهم إنه الأكتيفيتي أو الأماونت أو الفانكشن تبع هذا الإنزيم is compromised أو it has a problem. طيب شو بيصير في هاي الحالة؟ In this case, remember that RBCs depend mainly on glycolysis as a source of energy because RBCs do not have nuclei. RBCs do not have mitochondria. And Krebs cycle high is not there. So they have to have glycolysis to get their energy. فبالتالي, if this is one step that produces energy, that produces ATP. So there will be a reduction in the amount of ATP produced by glycolysis in the RBCs. Okay, and this ATP is needed for RBCs. Okay, specifically for the sodium-potassium pumps, and these pumps are important for maintaining the flexible or the biconcave disc shape of the RBCs. Remember يعني أهم شيء في RBCs شكلها اللي هو biconcave disc is in that way to be able to bend these cells once they pass through narrow blood vessels or narrow خلينا نقول النهايات اللي هي نهايات البلاد فيزلز. تمام، capillaries sorry once they pass through capillaries. فبحتاج اني اطعجها من النص. تخيل لو انها كانت سماكتها كبيره وواحده continuous زي ما هي على الاطراف زي ما هي بالنص، بيكون صعب انك تطعجها او ممكن تكسر معك اكثر. لما بتكون بايكونكيف ديسك يعني من الوسط رقيقه ومن الاطراف اسمك، فبالتالي بصير اسهل ان نطعجها وبتعطيها فلكسبيليتي، ممكن ترجع لوضعها الطبيعي لما ترجع على بلاد فيزلز اكبر. الصوديوم potassium pump important for maintaining this shape، وهدول البامبس طبعا they need energy in the form of ATP، والenergy is reduced due to this deficiency. Okay, so there would be premature death of these RBCs لأنه صارت الفانكشن تبعهم compromised. They also the amount of energy they have is reduced, so this would result in their premature death، وهذا بيؤدي إلى يعني the appearance of this hemolytic chronic hemolytic anemia. hemolytic يعني خلايا عم تتحلل وبتتكسر، يعني عم بتموت بسبب هاي الديفيشنسي. So now we're done with the regulation inside the cells. Let's talk about some inhibitors that may affect glycolysis, but they are from outside or exogenous sources. For example, fluoride, and this is important for dental students. Fluoride تمام inhibits a specific step in the pathway, which is for enzyme enolase, catalyzed by enzyme enolase, in which 2-phosphoglycerate is converted to phosphoenol pyruvate. Okay, but this happens in bacterial cells or bacterial enolase, not in our enolase. So there would be no inhibition of glycolysis in our cells once you use a toothpaste with fluoride, or you apply fluoride, for example, on the teeth of some of your patient's teeth, on your patient's teeth, etc., for as a preventive procedure. So also for if you drink water that is fluoridated، طبعا احنا المية هون عندنا في الأردن وفي بلاد كثيرة طبعا الفلورايد مضاف إلها بكميات محددة طبعا بهدف طبعا حماية الأسنان as a preventive procedure to reduce dental cavities and dental caries. The presence of this fluoride would inhibit this enzyme in the bacterial cells. فبالتالي it would inhibit glycolysis in the bacteria، يعني it would deprive these cells of ATP that is produced by the glycolysis reactions، فبتقلل الطاقة اللي بتحصلها. فبالتالي this would reduce the or this يعني may end up with reducing the multiplication and division of bacterial cells and results at the end in the prevention of dental caries. Okay. Another inhibitor of glycolysis اللي هو arsenic poisoning. arsenic poisoning، وهذا كان له أهمية في الماضي أكثر، لأنه يعني بعض أنواع الدهانات ومواد البناء المستخدمة كان يدخل فيها، لكن هلأ مش كتير يعني موجودة، بس in case بعض الناس ممكن يتعرض لها بسبب ظروف عمل معينة أو whatever. they may get يعني this type of poisoning. فطبعا تذكروا انه الارسنيك او الارسنيت له الارسنيك سوري له اكثر من نسميه من العناصر الانتقاليه. تمام. فمن احد الاشكال تبعته انه يكون بنتافالنت اللي هو الارسنيت. تمام. هذا الشكل specifically اللي هو البنتافالنت arsenic competes with this inorganic phosphates in which step of glycolysis in the formation of 1,3-bisphosphoglycerate from glyceraldehyde 3-phosphate اللي هي phosphorylation reaction that happens by glyceraldehyde 3-phosphate dehydrogenase. اللي هو oxidation reduction reaction associated with adding this phosphate اللي هو glyceraldehyde 3-phosphate dehydrogenase. طيب هلا شو اللي بيصير؟ It replaces this أو it competes with this phosphate. تمام. That is used as a substrate in addition to the glyceraldehyde 3-phosphate. there would be إيش؟ يعني it interferes with the completion of this step and the reactions would bypass this step to form 3-phosphoglycerate. يعني أنا ما راح أضيف فوسفات فبالتالي ما راح أقدر يكون عندي 2 phosphates in this molecule. I would just keep 1 phosphate اللي هي already on carbon number 3. تمام. there would be we would result with the formation of 3-phosphoglycerate. يعني there would be no formation of 1,3-bisphosphoglycerate. And notice that this step is associated with the formation of NADH. NADH is going to go into Krebs cycle and produces ATP and used to be to produce ATP. فبالتالي this would reduce ATP synthesis. يعني هي ما بتعمل inhibition direct للpathway نفسه بقدر ما إنها بتأثر على هدول side products that can be used to produce energy in a form of ATP later on يعني in the electron transport chain. Other form of arsenic اللي هو trivalent arsenic or arsenite. This one forms a complex and a stable complex يعني it cannot be detached easily with lipoic acid. Lipoic acid is the coenzyme needed for pyruvate dehydrogenase. Okay, so once it interacts with the thiol group of the lipoic acid, so it would inhibit the pyruvate dehydrogenase. فإذا عملت inactivation of pyruvate dehydrogenase شو بيصير؟ There would be no formation of molecules that can activate the Krebs cycle. There would be inactivation also of the alpha ketoglutarate dehydrogenase اللي هو عبارة عن enzyme in the Krebs cycle. Yes, so this would result in again interference with the production of energy by Krebs cycle and by electron transport chain later on. Okay, هذول طبعا from exogenous sources and they would result with neurological disturbances and they end up with death. طبعا حسب الامount اللي بتعرض له هذا الشخص والduration طبعا. With this we're done with the regulation of glycolysis. حكينا عنه سواء داخل الخلايا أو خارج الخلايا. كيف بصير بالجزئيات المختلفة وكيف بتنظم. حكينا عن الهرمونز كيف بتأثر على الباثواي إلى آخره. وريمبر ذات يعني most regulation إذا بنحكي عن داخل الخلايا كله عم بيحدث على الثلاث خطوات اللي حكينا عنهم from glucose to glucose 6-phosphate, from fructose 6-phosphate to fructose 1,6-bisphosphate, and from phosphoenol pyruvate to pyruvate. Okay.